mostly in Europe. I've worked over there, and also in North America. And also, of course, I live in India, so I work a little bit in India too. And the title of the talk is going to be in the applied domain of machine learning, graph algorithms, parallel supercomputing. Big data is a buzzword, anyways, and how we apply statistics in bioinformatics context. Much of the work will be in biomedical context, but you can relate very much to plant sciences, plant molecular sciences as well, because we are always going to talk about DNA. We are going to talk about gene regulation. We are going to talk about genetics, genetics of genes, these kind of things. I'm pretty sure it's very much dominant and predominant in the plant sciences as well. Okay, so having said that, let me, Professor Kapoor already introduced me, but to introduce myself, I wouldn't take too much words, but here are a couple of my, computer science and bioinformatics and biotechnology engagements. Some are studies, some are work. And I've also written 4 books. The 4th one, Machine Learning, is in writing, but 3 are already in press, and I'm gifting 1 book to the library of this department. Thank you so much. So much of the talk is going to be based on that book itself and also my upcoming book in machine learning. And the first discussion I'm going to have is about Sept. All the software that I'm mentioning is available on my GitHub page. Usually that's the web page where people launch the software for free, and they are available for free. All the source code is available for free. So it's essentially genome-wide massive sequence exhaustive comparison tool, such as for copy number variations. You must be knowing about these terms, right? Copy number variations, single nucleotide polymorphisms, insertions, deletions. If you want to do it for small sequences, it's a very easy task. Maybe you go to a web page and put your sequence and you get the result. But if you have a large amount of sequence, and not just for one sample, but several samples, several genomes, then the task becomes computationally intensive, and you know what the cost is if you go for collaboration. So why not use such freely available software that will do most of the work? So as you can see, these sequences are in the order of gigabytes, and the computation is all of the order of exponential format, the big O format. I won't get into the computational side of things, but usually the time goes for like this in exponential format. For small sequences, the time takes less, but for more larger sequences, the time goes up and up, and it becomes a really computational intensive task. And so when we want to do 2 genome comparison, we also need to map them properly so that we don't do redundant calculations. So we don't need to calculate the whole matrix. We do see where the replicas are, and we do not do a double computation. So how we format it is important, and how we fragment in the memory the sections that we want to compute is also important. So 1 genome versus the second genome, 1 genome versus the first genome, we don't need to do the double calculation here. The second genome versus the second genome, we don't need to do a double calculation. We just take care of the one that is relevant, because the other one is just a mirror image of it. And then what we see is that as the data size increases, the time increases linearly instead of going exponentially. So if you've studied maths and maths in class 12, you know what exponential equations are, and that's a big achievement, that we have reduced the time drastically based on the computation. And if we also make it in parallel, so if we use one processor, it can take such amount of time, but if we use multi-core processor, then the time gradually reduces significantly. So that's the software you can available, the Sept, you can use it for free. And then I talk about repurposing anti-inflammasome NRTIs, which is nucleoside this reverse transcriptase inhibitors. These are the compound molecules which have been used for HIV patients, okay, and which it has also been used for hepatitis patient. Okay, so the query is like if you know about how drugs are being formed, there's a lot of screening and a lot of process, a lot of money that's been pumped in, and lots of tests starting from rodent, mouse, and maybe to chimps and so on, and it's a really tedious process. So a strategy why we wanted to repurpose certain drugs is that we look at the biochemical pathway. If we see that there are certain segments of the whole biochemical pathway that can be common between one disease and another disease, and if you know the drug that is being used for one disease, why not try to see if that drug can be used for another disease? And before doing that, we can do this kind of filtering. So if we know that, okay, I have the data for all the patients, these you can buy it or get in collaboration with hospital clinics, and then we filter out the people who are having certain biochemical pathways that are being treated, such as hepatitis B or HIV, for which there are certain drugs that are being used. And then we look at the age group that we want to target, so we filter further that, and then we look at prior history of the person, and then once we have pocketed, segregated certain sections of the data, then we can see something like this. Initially we have millions of data, we filter it out. The raw data is in order of millions. This is 10-year data, 2006 to 2015. 10-year data, longitudinal data this is called. And then we filter out at every stage, and eventually we are able to get patients in certain category. And then what we essentially do is that we see that, okay, did that person also got cured for another disease, right? Yes or no? Okay, so in this case we see that no is high over there. Okay, and yes is in certain category. And then if we can see that, okay, there's a substantial amount of yes category or no category, just by looking at the data, we can also do some mathematical modeling like logistic regression. I'm not very sure if you know about it, but then if you at least are able to segregate the data to this level, it gives a certain clue that we can repurpose a drug for one disease to be used for another disease. Of course, we have to do further R&D, but at least we get some clue about can we do repurpose the same drug or with maybe some little modifications. Graph break. This is like graph science, graph algorithms everywhere, be it social science or anywhere. Graph algorithms, graph is like connections of different points, okay? And graph is a tool which is also used nowadays for, this is essentially, like, I'm one of the top initial person who are introducing these things. So we want to know networks of connections between diseases, between patients, between the genetic markers, between the variations. And here we talk about community and gene co-expression and linkage disequilibrium analysis. I'll talk about it in a second. So this is like about a community. If everybody knows Ensemble, for example, where we have the gene IDs by ENF. So here's an example of 3 genes, ENS ID on the right, and then all these numbers are structural variations, which can be a SNP, it can be indel, insertion deletion, it can be inversion, right? And the nomenclature is like we start with the chromosome number, the position number, and then what is the sequence in the reference genome. In human it would be like HG19 or HG19 point something, which is the latest version. For Arabidopsis thaliana, which is a model plant, there could be a certain reference number, and then you compare it to some other plant, or maybe it's a different strain of the same plant. Then you will have a different variant over there. And these variants that you're getting, they are connected to these genes. In a way, they are upregulating or downregulating these genes. And that's why they form community. And these are the communities which makes us very interested upon, because we know then where the molecular changes we need to introduce in order to make changes to the pathway and eventually to the phenotype. And so if we look using this tool called GraphBreak, which is freely available on GitHub, we can see all the variations. I mentioned them as SNP, single repeated polymorphism, but it could be something else as well. And then we have the list of genes. In human, there are roughly about 20,000 genes. and we can see where they are talking to each other. And in this way, we were able to find out 15 such communities, 15, 16 such communities where we can see they are talking a lot with each other. And they can be also, again, put back in this form of circles. And what do we do with that? One thing we can do is like take the genes and do a statistical overexpression or over function analysis. So this is like a list of different functions. We have immune system, we have diseases, we have metabolism in proteins and signal transduction. And wherever we see highlighted, for diseases as well, we have certain, within the disease subcategory and subcategory, we can see where those genes, those communities are leading to certain diseases, and then we can take appropriate action. And similarly, if you talk about plants, for example, you might have plants which get infected quickly with certain kind of viruses, and there are plants which don't get infected by the same kind of viruses. You can do similar kind of analysis. And there's also something called linkage disequilibrium, LD, which is like whenever there could be a true signal and there could be a false signal. A variant, which is referenced by RS. RS stands for reference SNP, okay, and the number. So a certain reference SNP could be affecting the overexpression or downregulation of a gene, but it could be that SNP is associated with another SNP which is a positive one. So we need to do LD variant linkage as well to find out which are the positive ones and which are just linked to them and not the real positive one. The enhancers and promoters and mini satellite, macro satellite, all these things, there are like many more things in genome than just the gene, which are regulating the whole thing. And we have to really pinpoint that, okay, this is the reference SNP which is affecting the upregulation of the gene and not the other one, right? And so even though things might show statistically everyone is significant, but out of these everyone, which is the one that is actually causing the effect? Another tool in this big data machine learning concept is about single cell technology. I think this is the buzzword in the last 5-6 years. Single cell technology comes with a major flaw, and that is that if you look at the data, you see a lot of zeros. So are they real zeros? Zeros, like these are the different genes, and whether they have been expressed or they are not expressed, or it was a fault in the technology. So we need to filter out these things, and that's why this basic tool, which is also available on GitHub for free, it looks at the RNA sequencing single cell data and it takes a k-nearest neighbor method approach, KNN method, and along with Bayesian statistics, it will do the imputation. Imputation is replacing the zeros with a more relevant number. So we don't have a sparse matrix like this, and I won't get into the Bayesian statistics. These are like very standard formula used in class 12 mathematics. But we need to identify which are the prior probability, which is the likelihood, and with the data. Once we have done that, we can apply that, and we can then make a 2D plot, how our data looks like. This is known as the dimensional reduction. So if you plot this 2D plot, it's usually called the TSNE plot or PCA plot, just to give you the idea about it. PCA, or the variant of PCA, is TSNE. So if you plot it before imputation and after imputation, there will be usually a big difference in single cell data that you get. So this technology, I wouldn't say that this is the only one. This is the one that I developed, but there are other tools as well in the market. Now coming to the core of this talk, which is deep learning, machine learning, genome-wide association study. Everybody knows about GWAS, for example, genome-wide association study, which has been really popular in the industry for more than a decade, actually 1.5 decades or so. Now, the GWAS does a multivariate analysis, and what we are now getting into is applying deep learning and machine learning, which is very popular these days, on the data. And I use the GTEx data for this. patient was used was heart attack. Uh, People who died out of heart attack. And this is how you look at, once you have compared a reference genome with your patient sample, in the best possible mapping that you do, right? For example, you must be knowing about BLAST, which is so popular, but there are now better tools. I would only say they're better, but there are many other tools than the BLAST, like BWA and all those things. Once you have used that for alignment, the best possible way, and then you look for the gaps. So the gaps could be filled up having a single nucleotide polymorphism like this, or it could be a large insertions or deletions. Okay? So the whole idea in machine learning is, I'm not teaching you machine learning, but you should know that the machine learning will work when you have numbers over there instead of letters. So you need to have an appropriate conversion method of converting this into a number. And I'm pretty sure everybody knows about multiple sequence alignment, MSA, right? So in multiple sequence alignment, we are doing the same thing. We generate a consensus sequence and look at all the sequences, how much different they are from the consensus sequence, and we get a score for it. So why not use the score as representing the variation of those insertions and deletions from the common consensus sequence. So this was proposed by me, and then we use standard algorithms like logistic regression, naive Bayes, support vector, random forest, and I also made my own algorithm called exhaustive deep neural network, which is like we vary the number of nodes and the number of layers. Usually deep neural learning network can take a lot of time, but it comes with very good accuracy. As you can see, only 100% accuracy. But it can be over learning as well, right? It will be overfitting the data. Once we have that, then we can have a qualitative impression of how good the data is. This is called confusion matrix. I wouldn't get into the details. A better representation is the receiver operating characteristics, where if you have this line above this diagonal line, then you have better confidence on your model. Why are we making this model? The whole reason is that once we have made a model that fits on the known data, that fits makes an impression about how things are, then we can use the model on unknown data, and we can make predictions such as biomarkers, right? Then we use the model for an unknown data or a new sample, I would say, and then we get a confidence for different positions, and we can relate it to, say, I got this position as an important position, having a good probabilistic score, and then we can look at the genotype of that okay, it is in chromosome 9 at this position, and it has a variant ID as well. So this would be a biomarker for, say, people who are prone to heart attack, right? Rather than looking at the whole genomic sequence and feeding it into our software, doing data analysis, we simply look at the biomarkers and we come up with certain confidence that, okay, this plant, Arabidopsis thaliana, has a trait to grow faster than many other wild-type species, for instance, right? Because maybe it has certain biomarker which tells that, okay, this plant is able to grow in just 3 weeks, whereas the wild-type plant takes a couple of months to grow. And that's like in your plant sciences context, but in the biomedical context, it would be like a human who got susceptible to certain disease, and he has more probability to get certain kind of disease. So biomarker research, this is like a really like $1 million or $1 billion industry.
How did you reach from multiple data sets to this variant?
Yes, sir. So from the multiple data set, I initially generated these variants by mapping. All the variants need to give a mathematical number for the machine learning algorithms to work. So to get those numbers, I do a multiple sequence alignment. I get the deviation of those scores. I replace these sequences by that number.
So in machine learning, what kind of question you are asking?
Yes. So the machine learning is usually supervised learning or unsupervised learning, broadly classified. They can find further subcategories. We are talking about here supervised learning, where we have patient sample data already for those who got, who died out of some heart disease or something. It could be some other trait as well. And once we got those genetic data and the final outcome data, we made a model by machine learning, so using these algorithms. ADA boost, gradient boost, naive Bayes, Bayesian, these are very standard algorithms. I won't get into the details. But I myself made a variant of the algorithm called exhaustive deep neural network, which is different layers.
So basically, in this supervised learning, you tell the algorithm that, okay, this is the patient, or these are the patients. Now find similarities between them.
Exactly.
That's it.
Yes, and we try to make a mathematical model for it, and instead of me trying to find that mathematics, there are already algorithms. These are the algorithms which tries to make that mathematical model and fits that